I'm going to go ahead and start, and just hopefully we'll have some people kind of pop back in as we're going. So first of all, hi, I'm Dr. Vicki Channon. I'm a teaching assistant professor in psychology and neuroscience. I'm also our director of undergrad research for psychology. I have a counterpart, Monica Gaudier Diaz, who is the director of undergrad research for neuroscience. I also teach a number of neuroscience classes and have like a human neuroscience background, and we work really closely together. So generally, if anybody is a neuroscience major and has questions about getting involved in research or Neuroscience 395, I'm also happy to answer any of those. I hope you all have had a chance to kind of look over the Sakai site and see all of the labs that are participating in this week's events. We've had great turnout at our synchronous session so far. And the way that it's going to work here is we have 4 different researchers who are going to tell us a little bit about their research, just kind of a 5-minute blurb about what the lab does. So Dr. Rebecca Stevens is speaking second, and she is going to tell us actually about 2 different labs. And so then after we have everyone tell you briefly about their lab, I'll open up some breakout rooms. Each of the researchers will go to a breakout room, and students, you can join them and ask questions or introduce yourself. And again, if you're interested in either of the labs that Dr. Stevens is going to talk about, you can go into that breakout room and ask questions. And that seems to have been kind of the most valuable part of this event, or these synchronous sessions for students, is that asking questions. So I'm going to ask the researchers to make sure you try not to go too far over 5 minutes, just to make sure we have plenty of time for that interactive piece. I am going to just get rid of my screen share, and we'll just go in this order here. So Zaire, if you would like to go first and talk about some of the research in the Family Journeys CoLab, that would be great.
Yeah, I can definitely do that. Let me just share my screen real quick. Okay. So, hey everyone, I'm Zaire Collins, and I'll be talking to you about the Family Journeys CoLab, which is headed by Andrea Hassong. So just like a little bit about me, I'm currently a senior at UNC. I'm originally from Winston-Salem, North Carolina, which is like an hour away. And I'm actually involved in a lab in a Psych 395 kind of role. And with Psych 395, you have 2 different options of what to turn in at the end of the semester. And I chose to do a review paper where I'll be studying substance use within sibling pairs. So just like a little bit about the lab. This is taken directly from our website. The Family Journeys CoLab is actually a collaborative research group that tends to focus mainly on areas of adolescent development, whether it's gratitude, substance use, identity formation, or social media. Dr. Hassong has actually made a lot of great programs kind of teaching gratitude within children. And as you can see down here, there's a lot of projects going on in our lab at the moment. So just some of them, the Real You study, which is kind of measuring mental health and substance use within college students. We also have Gratitude Conversations program and Raising Grateful Children, which is just kind of teaching parents how to teach gratitude within their children, how to facilitate conversations where children can kind of express their gratitude for anything that's going on in their lives. And the complex identity in today's youth in social media and identity is actually 2 of the projects that I work on in the lab. So the social media and identity, aka SMID, is actually one of our collaborative efforts with the Character Lab, where there's a survey given to high schoolers where they're asked to kind of judge a person based off of what they would post on social media. So these aren't actual people. We just kind of put up fake social media posts and ask the high schoolers to tell us what they think about them and rate them on a scale of 1 to 10. And we're actually in the process of coding all their responses at the moment. The Complex Identities in Today's Youth survey is actually just brand new. It started this semester. It's headed up by our grad student, Amanda, and it's meant to get at intersectionality within high schoolers. So they're kind of asked to rank their identities, describe their identities and how they interact with each other, and how comfortable they are disclosing certain identities to certain groups of people whether it be strangers or family members. And Amanda's research is kind of more centered around LGBTQ plus youth. So that is where she's trying to get at with her survey. And finally, my individual research project within the lab, which is where I'm studying the relationship between older sibling substance use on later born siblings. And just to kind of get into that, the reason I chose this was that most siblings tend to share the same home environment. If you share parents, then you both have similar genetic risk for either using substances or becoming addicted to substances. And if you share the same house and an older sibling is using substances, there's a chance that the younger sibling would have access to using the substance. And with a younger exposure to substances, there's kind of been research showing that you would have worse life outcomes later on in life. So whether it be a prevalence of mental illness or lower socioeconomic status, I was just kind of interested in studying that. And through my research, I've kind of examined a number of factors. So the gender of the siblings, which research has shown that brother-brother dyads actually increase risky behavior, which I thought was really interesting. The number of siblings that you have within a family could also impact how substance use would show up in that family. The court involvement of older siblings. So if a younger sibling is witnessing their older sibling go through the justice system, then that can have an impact on their own substance use. Hospitalization, kind of the same thing. Cultural values surrounding substances. So if there's kind of a culture where substances aren't looked on in a good light, then you're less likely to use substances anyways, regardless of if your older sibling uses them. And finally, the ages of the siblings and the gap between the siblings. So if there's like a 20-year gap between siblings, then what the older sibling does is probably not going to impact the younger sibling as much as it would if the siblings were about 2 or 3 years apart. And that is my presentation.
Awesome, thank you so much. Yes, and so I love when the undergrads come and present at these also. So Zaire might be a great breakout room to go in if you want to ask questions too about how she found her position and how she got involved. But yes, thank you very much. Okay, next up we have Rebecca Stevens, and she's again going to tell us about 2 different labs, the early brain development study, as well as the B lab, brain and early experience.
Yeah, so my name is Rebecca Stevens. I'm a research assistant professor in the Department of Psychiatry over in the School of Medicine. I'm also an adjunct instructor in psychology and neuroscience. And I'm going to be talking about 2 different labs. If you'll give me just a second to pull up one of them. Okay, so I'll talk first about the early brain development study. And this is a project within the School of Medicine, the Department of Psychiatry. Our PI is Dr. John Gilmore, and this is a study that is really looking at longitudinal development of both brain and behavior. And we have a subset of kids in this study that were born to mothers who had been diagnosed with some sort of psychiatric disorder, such as schizophrenia or bipolar disorder. And so that means that these kids have a slightly higher risk of eventually going on to also be diagnosed with one of those disorders. And so we're really interested in looking to see if we can identify early biomarkers of a later diagnosis. And so looking both at the brain and some early behaviors to see, is there anything predictive of these later outcomes? So here's a little bit about the study as a whole and the visits. Mothers were recruited prenatally, and then we had MRI scans right after birth, and then MRI and lab visits at 1 to 4 years, and then basically every 2 years. And we actually have 16-year-olds at this point, which is very exciting. This study has been going on for a while. We're currently deciding what to do with age 18 and how that would potentially work. And here I've included just a little bit about some of the data we collect. Like I mentioned, we're interested in these biomarkers of potential later diagnosis. So we have psychiatric history from the mother. We're collecting MRI data at every visit, and then a variety of cognitive and behavioral outcomes. These are just a few that I've listed here. But yeah, we have a pretty comprehensive lab visit to get quite a bit of data from our kids. In terms of how students are involved, typically when we bring on undergraduates to help out, they're helping with our image processing. And so basically you go from getting a picture from the MRI scan, how do you get that all the way to having data, like numbers that you can analyze and publish? And there are many, many steps in between. And that's where our students often help out. We have students helping with what's called brain masking. That's essentially just isolating the brain from everything else. Like you're getting rid of the skull and CSF and things that are not brain. And then there are various QC steps. And then we have some other options in terms of just study administration, some data entry. And you do have the opportunity to, if you want to, kind of set up your own study and analyze some of our existing data. We have some options for that. And then just a couple of other things I wanted to mention, COVID restrictions dependent, of course, there are potentially opportunities to observe study visits, both the behavioral assessments and maybe the MRI eventually. Again, some of this is dependent on capacity restrictions, but you'll meet with me and various other team members fairly regularly just to check in, see how things are going, and then have the opportunity to join the full lab and see kind of how a project that's this large and this extensive runs. So if you're interested in research, you can kind of get a glimpse at how a long-term study functions. And so if you happen to be interested in this, we don't have a formal application process. So you just need to email me. My email address I did not include on this page, which I should have done, but it was on the first slide. And I think it's also on the website. I assume Dr. Channon put that on there somewhere. And we do take students as either Psych or Neuroscience 395. We have volunteers, and we also do work with students who have federal work study. So if you have any questions, I look forward to answering those. And that is the first study I'm going to talk about. So if you give me just a second to switch over and pull up my B information. And then I will share my screen again. Okay, so the B study is another project that I work on. And this is through Frank Porter Graham Child Development Institute. And we have a couple of PIs for this study. There's Dr. Kathy Proper, who works through FPG and also the Department of Psychology and Neuroscience, and Dr. Sarah Short, who's actually at Wisconsin. So it's a sort of multi-institution project. And this kind of summarizes exactly the big picture of what we're looking at. So we're looking at how prenatal factors and also postnatal experiences influence infant behavior, cognition, brain development. And this says infant in the official description, but we are following kids up through toddlerhood and hoping to continue through early childhood, middle childhood, and potentially beyond. So to summarize the visits in this study, similarly to the other one, we started prenatally. This is when moms were recruited. Then there was a 2-week visit with an MRI, and then we've had 6-month, 18-month, and we just started our 30-month visits actually planned. We have 36-month, and then of course, dependent on later funding, we're hoping to continue following these kids for a while. And then here's some of the data that we are collecting. So we have MRI visits twice so far, so 2-week, and we just started 30-month. And we, at every single visit, collect a variety of information in terms of parent report surveys. We get some child behavior, parent-child interaction, looking for things like maternal sensitivity. We're really interested in sleep. So we actually have kids wear sort of like a glorified Fitbit that they wear around their ankle for a week that measures activity level and sleep. And so we're collecting a whole bunch of information to get kind of a full picture of the context and how these kids are growing up. In terms of how students are involved in this study, we have our undergraduates working kind of across the board through almost every single aspect, especially with data collection. So depending on the study visit, because each one is a little bit different, we often have students actually basically running the visit from start to finish in terms of contacting our participants, scheduling the visit, actually running the visit, and then doing all the post-visit everything. Some others, we have students who come in and just assist with either assessments, or we do have students helping out with our MRIs. And then of course we have a variety of other roles in terms of data entry. We look at medical records. I mentioned actigraphy editing. This is the wrist or the ankle bracelet that they wear that measures sleep data. So we have a variety of roles for students. And then in terms of everything else you can expect, as I mentioned before, we're a multi-institutional team and it is a very large group. So you'll get some experience working with a very, very large team. We have very regular meetings. So you will meet at least weekly with various teams on this project. And over the past couple of semesters, we've also had journal club and professional development meetings. And so journal club gives you the opportunity to practice reading scientific articles and analyzing those and then discussing them with other students and also staff, grad students, postdocs, and sometimes PIs. And then I'm particularly proud of these professional development meetings. We've been able to hold usually a few per semester, and we have a variety of individuals from kind of all levels of the study joining and providing information about what their experiences were, guidance for applying to grad school, possible directions after undergrad. I mean, you name it, we can probably talk about it in one of these meetings. So if you are interested, we are currently accepting applications for the B-Lab. This is a formal application process. I know this is a long link, but you can pause this video later and hopefully catch that. But I've also put a QR code here that you can scan. It's going to take you to this website where there's a link to a Google form if you're interested in applying. Similarly to my other study, we take on students as 395 volunteer or work study. So there are some options there. And that's it. Thank you.
Thanks so much, Rebecca. And if you want to send me that QR code or the link, I can also put that in the listing and the asynchronous stuff for the B-Lab. And I've had a number of my students that have worked in the B-Lab that I've spoken to, and they all have wonderful things to say about the training that happens in that lab. So, okay, let's see, where's my slide? Next up, we have Mackenzie Mitchell, who is in Jessica Cohen's lab.
Yes, hello. Let me share. Can you see this? Yeah, okay, great. So like Vicki said, my name is Mackenzie and I'm a third-year graduate student in cognitive psychology and I work with Dr. Jessica Cohen. And I'm gonna, I guess the general science that I do is developmental cognitive neuroscience. So I wanna kind of break that down for you first. So developmental, meaning that I work with kids, not that I work with embryonic development. So I'm not looking at like cortical development of like the neural tube. But this would be like through infancy up into early adulthood. And my particular area of interest is around puberty. And then cognitive, which includes things like language, attention, memory, learning, and executive function. And I would say that our lab is interested in a lot of things and covers a lot of these domains, but executive function is kind of the primary one. And then neuroscience, meaning that we study development and cognition through the lens of the brain and how the brain changes as kids grow up. And this is just kind of a classic image of a study showing like cortical brain development from kids 5 to 20. And so the kind of research questions that I ask and that my lab is asking are things like, how is the brain involved in self-control? And when I say self-control, we do look a little bit at emotion regulation, but a lot of what we're looking at is like motor control. So here, like in the Squid Game, I can see Elena reacting to this. So I'm gonna assume that Elena has watched this. But in this game, if you haven't seen it, it's red light, green light. And so they have to run at a certain cue and then stop themselves from running at another cue. And so your ability to kind of control your movements as you go through this game contributes to your success. And in this case, your life, but that's not really how we're studying it. So another example for kids could be like you're crossing a crosswalk and you need to be able to take in perceptual information from your environment and know when it's safe to walk and when you need to stop. And then another type of self-control, which there it is or inhibition is one of the words we use for this, is stopping yourself from distraction. So kind of keeping your attention focused on what you're doing at hand. And there are individual variability in all of these things that kids do. And so keeping your attention on one thing could be easier for some kids than for others. For example, children with ADHD have a harder time with this, and that's one of the populations we study in our lab. Other kinds of questions that we ask are things like, how does brain function change as children grow into teens and adults? And I'm not going to break down this image, but basically it's from a task of motor control or self-control. And we can look at how these maps of activity kind of change as kids go through development. And then another kind of question that our lab looks at is, how does brain function kind of flexibly change in different contexts? So your brain might look and be acting in one way while you're daydreaming, and then it kind of changes and does something else when you're focusing or working really hard. And so how does your brain make that flexible switch between those different states? And again, that's easier for some individuals than other individuals. So what does it mean for kids who are really good at that and others who really struggle with it? And so our research approach is to have kids come into the lab and we ask them lots of questions about their behaviors and mental health. And then we also ask them to play computer games, tapping at things like self-control or other executive functions or like reward learning. And we also have them go into an MRI machine. So this donut-shaped machine, kids lay on a table, and I don't know if you can see, but she's holding like basically a video game controller in her hands. And so then we would slide her into the MRI, and then we can take pictures of her brain function while she's playing games. And so we can get out a picture kind of like this. So this is a brain this was taken during like a task of reading. So these are the regions that are more active during reading than resting. I am not going to go into this. I have this like nice little graphic talking about how we kind of combine neuroscience and mathematics and to look at something called functional brain connectivity. And I'm happy to talk more about this in the breakout rooms if anyone is interested. So I'm super excited about it, and our lab does a lot of it. But for the sake of time, I'll just say that we have one study that we're in active recruitment for right now. It's called ADHD Brain Map, where MAP is an acronym for Maturation Across Puberty. So we have participants between the ages of 10 and 12 coming in, kids with ADHD and some kids without ADHD. This is Jess Cohen getting into the scanner. So you have kids with ADHD and not ADHD, and we have them coming in for 3 years. So we're tracking them longitudinally across puberty. And RA roles could be prepping for visits and actually like data collection, so running the subjects and interacting with kids and families. And then of course, data management, literature reviews. And then after you've been in the lab for a little bit, we have opportunities for doing research projects where you could pair up with a grad student or postdoc. And I wanted to include this fun picture. We were delayed because of COVID, but we are finally up and running again, which is really great. And so this is our PI, Dr. Cohen, prepping, like piloting some of the scans for us. And of course, here's our lab and the link to how you can get involved.
Awesome. Thank you very much. Y'all are doing a much better job at staying on time. The very first session, it was a little bit of a disaster, but I've been trying to like remind people a little bit more now. All right, and then we have last, but certainly not least, Elena. Let's see, Pedrascu. You're gonna have to help me with that one.
Great. All right, thank you. All right, so not to end and make this a disaster, I am going to try to go relatively quickly through my slides, but obviously if there's at any point that you want more information about, just ask me more about it in the breakout room. All right, so my name's Elena. I am a third-year graduate student in the Behavioral and Integrative Neuroscience program, and I am in the CoLab, so Cognition and Addiction Behavioral Neuroscience. So just a little bit of background about why we do what we do. So we primarily study intermediate phenotypes of alcohol use disorder. And so, as we know, this and substance use disorder in general is extremely prevalent. This is just showing that in 2019, about 15 million people were diagnosed with alcohol use disorder. And so one way to kind of target this problem is to maybe prevent it from happening in the first place. So we look a lot at adolescence and specifically patterns of drinking that are considered very like heavy alcohol use. So binge drinking is one of those patterns. And we know that the earlier age of onset of alcohol use, as well as these heavy patterns of drinking, really increase that risk of future development of alcohol use disorder. So we study various intermediate phenotypes, such as impulsivity as a behavioral characteristic. We primarily study behavioral flexibility. We have 2 computerized tasks that kind of measure this. And then also we, similar to what Mackenzie was mentioning, so I won't go into depth about that, but we look at activation of various brain regions, as well as the communication between those brain regions. And so I'll just go over some projects that some current lab members, as well as myself, have been working on, and some future ones. So Dr. Elton's a research associate professor in our lab, and she looks a lot at how like early adverse childhood experiences and family history of alcohol use disorder kind of interact with impulsivity and predict future alcohol use problems. So she does a lot of longitudinal work. And so this current study that she has been looking at collects all of this different information, some of what I just mentioned, and then also does functional magnetic resonance imaging. So the participants, these are college-age students, and so they do a couple tasks that measure impulsivity and inhibition. And then she looks at activation of brain regions while they are doing this task, and then follows up with them to look at alcohol and other substance use patterns, as well as other measures. Maddie Robertson, one of my colleagues, she's a dual student, so she's also in the clinical psychology program. And so her study, along with mine, they're like sister studies that really look at binge drinking. So we have adults aged 22 to 40. And so we look at what basically the persistent effects of binge drinking during adolescence have on behavioral flexibility, as well as using different neuroimaging methods to see what happens in the brain. And so her neuroimaging methods include EEG and alternating current stimulation. So one form of behavioral flexibility is habitual versus goal-directed behavior. And so you can think of that as if you have a certain cue, like, oh, it's time to eat lunch, then just your routine might be to go get food and then whatever that reward is. And so this habitual behavior can be positive, but it can be negative in the case of alcohol use disorder, persistent use of alcohol or another substance despite negative consequences. And so she uses EEG. So we place these electrodes all over the brain and can measure brain activity to see what this like basal resting state brain activity is for various participants, and then use that information for when she uses transcranial alternating current stimulation, where we place electrodes over the dorsolateral prefrontal cortex, which is very important for goal-directed behavior, while they're doing this task that measures behavioral flexibility. So seeing how this manipulation can change or alter behavioral flexibility. Okay. And then the sister study that I'm currently working on, so aside from looking at habitual versus goal-directed behavior, another proxy of flexibility is attentional bias. And so you can just think of that as if you're presented with a bowl of carrots, but then in the distance there are these delicious chips, how much does that stimulus really capture your attention and bias it towards looking at that versus the goal, which might be eating the carrots. And so in addition to that, they do that task in the scanner. And so we look at activation of brain regions while they're doing that task, and then also looking at communication between different brain regions involved in addiction while they're not engaged in any task. And then the last thing I'm going to mention is I have a pilot study that I'm going to be starting this upcoming spring. And depending on funding, hopefully that will continue over the next 2 years. But so one idea is, once we know these intermediate phenotypes, can we intervene with certain interventions and prevent alcohol use disorder? So I am very interested in mindfulness meditation. And so the study that I will be doing basically looks at first-year college students who have a history of adolescent binge drinking and implement this mindfulness meditation course that's specifically designed for emerging adults, and look at behavioral flexibility. So attentional bias is one of them, collect inflammatory markers, and then so pre and post training, and then follow up with them to see any persistent effects on alcohol use. And so aside from the study that I'm currently working on, it's nearing an end, but I do have some opportunities available for students if interested. And then also for the mindfulness meditation study, since that will be, going off the ground anew next semester, there are some potential opportunities for that. And for 395, we definitely welcome students to do 395, but we do like that they volunteer for at least a semester before that. And that is it.
Okay, awesome. All right, that was great. Okay, I am going to once again share my screen so that y'all can see the different numbers here. Yeah, so I'm just going to open 4 breakout rooms, and you can see the numbers here, 1, 2, 3, 4, who will be in which breakout room. I'll stay in this main room. If any students have any questions, general questions or anything, I'm happy to answer them. I'll also just keep this screen shared in case you need to pop back in to see which other breakout room you might want to join, but you can move amongst the different breakout rooms as you get your questions answered or introduce yourself and want to move to another one. I know some of our researchers have to leave at 1. I also need to leave at like 5 after 1. I can keep the meeting open a little bit longer than 1, but not much. But otherwise, I'm going to go ahead and just let you all kind of move amongst the breakout rooms. I do have a survey that we would really love for the students to fill out about the event, both the asynchronous information that you may have accessed this week, as well as any synchronous sessions that you've attended. And so I just put the link to that survey in chat. We want to make this event each year exactly what is most helpful for students. So any feedback you can provide would be great in terms of what we should keep or do differently next year. I think that's it. If you have an old version of Zoom and you find that when I open the breakout rooms, you can't go to where you're trying to go, just let me know and I can move you into whichever room you want. You can either just unmute yourself or you can put it in chat and I can move you into whichever room you would like to move to. All right, I am going to open 4 breakout rooms. Everybody understand what they're doing? Awesome. Okay,